can I have your attention, please? Boy, that was quick. Thank you. Good afternoon. I'm Alan Winter, President and CEO of Genome BC, and it's great to see you all here. Welcome to the second annual Don Ricks Distinguished Keynote Address. And today we recognize Don Ricks, one of the co-founders of Genome BC and our chair for 4 formative years. So Don was a physician who saw the need in his practice for better diagnostics and founded a system of clinical labs which today is known as Life Labs across Canada. He was also physician to Michael Smith, who was the UBC Nobel laureate, who convinced him that genomics was an important part of the future. So to quote Haig Farris, who was also on our board at that time, Don Ricks was a man of action, a man of vision, a man of compassion, and a great Canadian. He was a great entrepreneur, a committed angel investor, and a generous and effective donor to many causes. Don was quick to support young entrepreneurs and educational initiatives, and we may hear more about that today. Where others saw problems, Don saw opportunities. His cheerful, constructive contribution to every organization he supported was an inspiration to everyone. His legacy of commitment will influence our community for generations. So as a result, the Genome BC board, and there are several board members here today, and we appreciate your presence, the Genome BC board decided that one way we could recognize Don Ricks and his legacy is to establish this annual keynote address as a way to encourage and inspire our community. Our first guest last year at our 10th anniversary was Sir Mark Walport, President of the Wellcome Trust. And tonight we're especially honored to have Dr. Leroy Hood with us as our distinguished speaker, and let me introduce him to you. So Lee Hood is president and co-founder of the Institute of Systems Biology in Seattle, Washington. And in fact, I think in the notice we sent out a background to Lee Hood. So let me just pick up a couple of things which, again, links with what I was mentioning about Don Ricks and helps us with understanding the context for this evening's talk. So Lee Hood is a pioneer in the systems approach to biology and to medicine. His research has focused on the study of molecular immunology, biotechnology, and genomics. His research career began at Caltech, where he and his colleagues developed DNA gene sequencer and synthesizer, and protein sequencer and synthesizer. So those 4 instruments paved the way for successful mapping of the human genome and led him to receiving several prestigious prizes, and I won't go through all of them. A pillar in the biotech field, he has played a role in the founding of more than 14 companies, including Amgen, a small company, and Applied Biosystems. In terms of recognition, he's a member of the National Academy of Sciences, the National Academy of Engineering, the Institute of Medicine, and one of only 10 people in the world to be accepted to all 3. And that's really a background to the interdisciplinary nature of Lee Hood. He has published widely, including a popular book on the Human Genome Project, The Code of Codes. He's received many awards, including the Lasker Award and the Kyoto Prize, and 17 honorary degrees, and holds over 30 patents. Now, after the address, so that we're next going to listen to Lee, there's to be a question and answer period moderated by Dr. Brad Popovich, our Chief Scientific Officer, and then a reception outside. And then you may want to ask him more about what really drove him towards science. So you can ask him, for example, was it growing up in Montana? Or spending summers at a geology camp with his grandfather in the Beartooth Mountains, which I think was also Montana? Or was it playing football at the Rose Bowl? Or was it playing the oboe? Or climbing the mountains of the West Coast? All these questions you can ask him, but first we're going to ask him to give his address to us. So Lee, it's a great pleasure to welcome you to the podium.
Well, thank you very much for that kind introduction. What I'd like to do today is talk about P4 medicine, which I think is a revolution in medicine that's going to change not only the practice of medicine, but how we think of medicine as well. And in fact, to give you an idea of how big a revolution this is, my contention is that in 10 years, many of us as individual patients will be surrounded by a virtual cloud of billions of data points, and we'll have the wherewithal to reduce that enormous data dimensionality to simple hypotheses about health and disease. And the critical thing is we will do that for each of you independently. The essence of this P4 medicine. Now, a question you can ask, which is really a fascinating one, is why do we need all of this data? And there's a very simple explanation. And the explanation is because biology is incredibly complicated. And the next question we can ask is, why is biology so complicated? And it turns out that there is really a simple solution to that. And it's the simple fact of Darwinian evolution. Darwinian evolution isn't linear and progressive, rather it's random and chaotic. It builds on what succeeded in the past. There is no law of Occam's razor in biology. And in fact, what it reminds me very much of is this Rube Goldberg cartoon. Here is a device that has 14 little gadgets that together collectively allow Goldberg to cool his soup. And that reminds me of biological complexity. So the question that's really interesting is, how would you study that device so you could understand how the soup is cooled? So number one, you'd need to know all of the components present in that device. Number two, you'd need to know how they're connected. And number three, you'd need to know their dynamics and how they operate. That is exactly what we need to know in biological complexity, and that's exactly what a systems approach to disease that we'll talk about is really all about. Now, one of the interesting questions is, with all of this complexity, and I started thinking about this in the early 70s, how do we go about deciphering disease, deciphering health? And in fact, when I was a young assistant professor at Caltech, I read a book that really transformed my life in many ways. It was Thomas Kuhn's The Structure of Scientific Revolution, and what it was about was paradigm changes. And it pointed out that all of the non-giant step changes in science came as a consequence of paradigm changes. And it pointed out that one of the common features of the paradigm change was enormous skepticism and difficulty in actually having it be accepted. So I had the good fortune to really participate in 5 paradigm changes that led us up to this thing we're going to call P4 medicine. And I'll very briefly tell you what they were. The first you've heard, it was bringing engineering in the early days to biology, developing the 4 instruments that you heard about, but the essence of what those instruments made possible through automation and parallelization, integration, was high-throughput biology, the ability to generate lots of data. The second paradigm change actually came about as a consequence of being in the process of developing the automated DNA sequencer, when I got invited in the spring of 1985 to the first meeting ever on the Human Genome Project. Bob Sinsheimer at Santa Cruz was debating with the idea of starting an institute there, so we invited 12 of us to go and consider this as a proposition. So we debated for several days and came to 2 interesting conclusions. One was it was feasible, but the other was that most of the biologists were really going to oppose this idea. And in the beginning, it was more than 90% that opposed it for lots of interesting doctrinary reasons. Now, again, in developing the automated DNA sequencer, it brought me to the third paradigm change, And that was the realization that biology had to become cross-disciplinary, that because the leading-edge problems in biology needed the development of new technologies and computational tools, you had to create cross-disciplinary environments. I tried to do this at Caltech. The biologists were against it. Bill Gates made it possible for me to go and do it at the University of Washington, and that department was absolutely a spectacular success. What I planned to do, and the original dean agreed to, was that I would build, after 4 or 5 years on top of that cross-disciplinary department, a systems biology feature. And unfortunately, the dean that agreed to that died in an avalanche in the Himalayas, and the next dean had a very different point of view. So I ended up, after a few years of trying, actually resigning from the university to start the Institute for Systems Biology to be able to build this new approach, experimental approach to complexity. And thinking not only about biology, but about medicine as well, then the fifth paradigm change, which is P4 medicine, came to the fore. Now, what a systems approach to biology and disease is really about are these 4 fundamental pillars. One is the idea that biology is an informational science. We'll say a few words about that in a moment. The second is this holistic systems approach to dealing with complexity. The third is every hard problem demands new technologies so you can explore new dimensions of patient data space, in the case of medicine. And finally, you need the analytic tools, computational and mathematical, to be able to deal with all of that information. So what about biology as an informational science? I'll make 3 points. Number 1 is there are 2 types of biological information. There's the digital information that's present in your genome, and there's the environmental information that comes from outside the genome. The 2 of those types of information come together to create the phenotype of the organism through development, through physiologic responses, through the initiation and progression of disease. So the next interesting question is, what connects this information with the phenotype? And of course, there are 2 informational structures in living cells for doing that. One, of course, are the biological networks that capture, transmit, and integrate, and finally pass information on to molecular machines that are really the executors of function. So the integration of networks with molecular machines and the dynamics of how they carry out their function is one of the major purveyances of systems biology. Now, the final point I would make is biological information is hierarchical, going from DNA to RNA to protein, all the way down to individuals and populations and finally ecologies. What we know is the environment impinges on every one of those levels of information and modifies the digital signal. That means if you're truly to understand the system at one of those levels, say cell division at the level of cells, you have to capture the antecedent information back to the DNA, and you have to carry out a process of integration which does nothing more than explicate the environmental signals that have been fed in. It's only when you understand environment plus genomics that you can really gain a deep understanding of these biological systems.